Assalamualaikum, selamat pagi, Dr. Irfan. Good morning,
Good
Prof.
morning. Good morning, everyone.
Ini kemarin apakah ada Nadia yang mau jadi moderator atau siapa kemarin?
Hello, Prof. D. How are you?
I am fine, thank you. I hope we're supposed to have a big rainstorm that comes right in the middle of this talk. So if I suddenly go away, you know that we have lost electricity or internet.
Yes.
But
okay, I hope not.
yeah, me too.
Thank you. How are you, Prof?
I am doing well, thank you.
Oh, thank you very much. So good evening. So we are very sorry that we cannot come to America for the APS meeting. Like I said you before, it's exactly the Eid al-Fitri of the time.
Oh, I know. Bad timing.
Yes.
Bad timing.
So sorry. So actually I was planned to go, but suddenly I realized that this time is same as it's impossible for us to go.
Yes. Yeah, that's a family time. Very important.
Yes, of
to
course,
be with family.
Once a year. So do you have a plan to go to Daegu, Korea on next October for FAUP's meeting?
I had not thought about that. I will be going to Pan American meeting in November down in Chile.
Yeah. Okay. Yes, we have FAUP's Federation Asia Oceania meeting in Daegu, October 1 until 3. I hope I can see you there.
And where will it be held?
In South Korea, Daegu.
Okay.
Maybe I can share your the link for that meeting. And thank you for your sharing for WhatsApp number. So I try to make you as in my contact. So make us easier to do that. And also probably that if I have your permission that we will share also this link to our Indonesian Physiology Society meeting for also following your lecture to this morning. Is it okay for you?
Certainly.
Okay. Thank you. Okay. Maybe Dr. Budi, siapa yang mau jadi moderator, Dr. Budi?
Mahasiswa bersedia kemarin, Dr. Irfan?
Oh, yeah, of course. Good one. Okay. Good one. So
mungkin di dalam ruangan
ini
besar.
Prof. Dwi, it's better that you know that we have a policy that doing the hybrids after the pandemic era. So actually we have 240 students, but some of them doing in offline in one class, and some of them doing in online with Zoom meeting. So you can see the mainstream. Maybe we thought you can share the mainstream with for the class. The block. Yeah, okay, you can see that.
There we go.
So, okay, and the moderator is also come from the student, probably. Okay.
Okay.
So
you can see my screen now.
Yes, of course. Yeah. Thank you very much, Prof. D. Okay. Prof. D said I will also follow your lecture, but I give a chance for the student to control this meeting. Okay, thank you, Prof. D. Okay, you may start, student.
Assalamualaikum. Excuse me, I'm sorry, but I'm still loading to my Zoom, so I will be talking from here first before joining via my Zoom. Apparently, my Zoom is loading.
Your voice is not loud enough.
Yeah. Can you hear me now?
Okay, good one.
Yes.
Okay, first of all, I'm sorry. I'm still trying to load into my Zoom, so I will be talking without camera first, but I will be trying to join via Zoom. I have the class right now. Shall we start?
Yes, fine with me to start.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. And good morning, all the lecturers of the Medicine Department of University of Sriwijaya. I'm Nadia Ratnariana from class Alpha, and today I will be acting as our lecture section moderator. For today's special class, we're also having an equally special guest, Professor D. Onglop Silverthorn. Good day, Professor Silverthorn. Thank you for joining. Yeah, thank you for joining in our class today. It's such a pleasure to see you. How are you faring today?
We are doing well here. We're about to have a big rainstorm.
Okay, I'm very glad to hear that. So Professor Silverthorn currently works at the Dell Medical School at University of Texas at Austin. She's also the author of various books, one of which is a book that may have assisted everyone here, including me, in studying physiology. That is Human Physiology, an integrated approach. What makes this book special is, as mentioned in the title, it is an integrative approach. It has integrative approach towards physiology. Now, allow me to welcome Professor Silverthorn to officially deliver her lecture. The screen is yours, Professor.
Wonderful, thank you. And can everyone see it's a picture of your beautiful bridge there in Palembang that I took when I was visiting there in 2010. So today we're going to be talking about my favorite topic, which is physiology. And it's an important topic because it crosses many different fields, but it is particularly important for you as medical students to know about it because it has a tremendous role in clinical medicine. So I thought I would start by asking you quickly to think to yourself, if you had to pick an organ system, which organ system do you think is the most important? And I listed some there. So think about that. And as it turns out, it's really hard to choose because they all work together, as this figure shows, that each of the systems is able to influence the other systems. I think many people would say the nervous system is the most important because if the brain goes down, then the rest of the body goes down. But there is a lot of overlap between all of the systems. And so that is what we will talk about today. One of the things that makes studying physiology a little bit difficult is that we study it in a linear fashion because time is linear. We start our course with something, perhaps cell physiology, and then we go into the different systems one at a time, as you're seeing here. And then at the end of the semester, we have covered all of the systems, but we covered them in a linear fashion, and physiology is integrated, not linear. So homeostasis and body function then requires the integration of multiple body systems. And your challenge as students of physiology will be to make that integration happen. And one of the ways that we are starting to think about it is to use what we call core concepts. These are the big ideas of physiology. They are patterns that repeat over and over in different systems. So if you learn what the big ideas are, then you can recognize them each time you come across them in a new system. So we will start with core concepts, then we will look at homeostasis, and then finally, we will see how the core concepts in homeostasis become so important in pathophysiology because people are integrated systems. We may not study physiology in an integrated fashion, but people have physiology that is highly integrated. So once you've got the organ systems down and have studied all of them, I think you will find it easier to see the core concepts and how they integrate. So the core concepts we will talk about today, we will start with structure, function, relationships. Then we will look very briefly at energy, gradients and flow, communication and information, and homeostasis. And then finally, how all of these integrate together into a functioning body. So structure and function has many different aspects. One is to think about it at different levels of organization. We can think about the body as a whole, or we can think about a system such as the cardiovascular system. But within that system, we have organs and tissues and cells and molecules and even ions and atoms that are important. One of my favorite core concepts is the one that we call compartmentation, meaning that the body is divided into many different kinds of compartments. Here you see a picture of a cell, and it has compartments within it. The cell itself is one compartment, but the Golgi or the nucleus would be compartments within the cell. The body fluid compartments are also then, obviously, because it says that, compartments. So what you see in this picture is the yellow cells. The purple is the interstitial fluid. And then you've got the plasma that has the liquid matrix, and within it floating are more cells, the blood cells. And then finally, our biggest compartments are the internal and external environments. So our body is the biggest compartment we have, and there is a constant exchange of material between our body and our external environment. And because of this, we say that the body is an open system. One way to look at it that is more schematic is to this picture that you see, where the cells are at the center, and then surrounding them is the extracellular fluid, or ECF, that's the purple. And the dividing boundary between these two compartments is the cell membrane in dark purple. And it is the extracellular fluid that is constantly exchanging material with the external environment. This is what we call a box diagram, and it is a schematic way to think about the compartments, but it's very useful. So on the left side in white, you see the external environment, and separating the external environment from the internal environment is an epithelium of some sort. And an epithelium is a cell layer. So anything crossing from the external environment into the body must go through two cell membranes. And then you see the extracellular fluid in purple, and then the single cell membrane that divides the intracellular fluid from the extracellular fluid. Remember then that everything that enters or leaves the body must first cross an epithelium before it can get into the extracellular fluid. These epithelia, in some cases, are passive, but in other cases, such as your digestive tract, they actually help regulate what enters and leaves the body. This is one of my favorite pictures because people seem to think that if it's in your body, if it's inside our skin, that it must be internal environment. And what this figure is showing you is that not all of the internal organs are internal to the part of the internal environment. So the word we use is lumen. This means the inside of a hollow organ or tube. And so where you see white in this figure, what we see is that the lumens of many of our organs are actually external environment. like the hole that goes through the middle of a bead. The hole goes through the bead, but it is not part of the bead. And I think our digestive system is probably the best example we have for that, because the internal environment is not in the lumen of the digestive system. This is how we can have pathogenic bacteria such as E. coli living in our large intestine without making us ill, unless they get through the wall, the epithelium, into the true internal environment. So that was compartmentation. Now, the other things that we have to think about for structure function are shape and size. On the left, you see the villi of the intestine, where the shape and size can actually change the surface area. So that idea of surface to volume ratio. And then on the right, there are compartments again, but notice that the wall, the epithelium between the plasma and the interstitial fluid is a very thin, flat layer of cells, the endothelium. And it is thin and flat with loose connections to allow very rapid exchange between the plasma and the interstitial fluid. We also have to think about the physical properties created by cell-cell junctions. Are they tight junctions that prevent movement across the epithelium? Or are they leaky junctions that allow some or significant movement? We also have to think about the matrix outside the cell and the way the cells are anchored to the matrix. So a cell matrix junction. And then we also have to think about mechanical properties of our tissue, such as elastance and compliance, which become very important particularly when we are thinking about pulmonary function. Energy is another one of our core concepts. And to simplify it, we're basically dealing with two types of energy. We have the potential energy stored in chemical bonds, and those are all of our biomolecules. We have fat and protein with lots of energy in the chemical bonds, and then ATP as our most usable source of chemical bond energy. But we also have potential energy that is stored in concentration gradients, electrical gradients, and electrochemical gradients. And so understanding the resting membrane potential of the cell, it depends upon understanding these electrochemical gradients and the potential energy that can be stored within them. Another theme, gradients and flow. We have many different types of flow throughout the body. We have the bulk flow of fluids. Air is a compressible fluid. Blood, because it is based on water, is a non-compressible fluid. But engineers consider them both fluids and discuss how the fluid flow takes place along a pressure gradient and is altered by the parameters such as the viscosity of the fluid that is flowing and the resistance of the vessel through which it's flowing. Then we have flow down electrical and chemical gradients, and that would include both across the cell membrane and through the interstitial fluid, as you see here, with a chemical being released from one cell and moving by diffusion down its concentration gradient to the neighboring cells. And then we have membrane transport, moving things across membranes, either through channels or on transporters. And sometimes the transport takes place down the chemical or electrochemical gradient. But in other instances, the cell is able to use energy, usually from ATP or from another molecule, to push a substance against its gradient. At the molecular level, the big theme is protein binding. Proteins bind to ligands, and there are many different examples that you've seen, both enzymes that bind to substrates. You have transporters that bind to their substrate that they are transporting. And you've also got simple protein binding to ligands for various other functions. So, for instance, this is the oxygen hemoglobin saturation curve showing the binding of oxygen to fetal hemoglobin compared to the maternal hemoglobin. And so we have to think when we talk about protein binding about specificity, competition, and saturation. Here's another example from the renal system. With this case, the protein binding is binding glucose binding to a transporter that is causing its reabsorption across the wall of the proximal tubule. And you can see then that you have saturation, and you show the relationship between concentration, the red dots, and the transport rate. So here's to sum this up in one example. This is a figure that shows the total oxygen content of arterial blood. And we know that oxygen occurs in two forms in the blood. It is dissolved in the plasma, the gold on the left side of the screen, and it's also bound to hemoglobin. And what we see are two of the principles that we just talked about. The physical properties of the tissues is indicated by the red box. So we're looking at the resistance to flow, down gradients. We look at lung compliance, the importance of surface area, and also the importance of diffusion distance. So if there's excess interstitial fluid, as with pulmonary edema, then that is going to alter the function. And then on the right side, we see the protein binding. You just saw the curve that we looked at for oxygen and fetal hemoglobin. So these themes show up, and it's easy then, once you are aware of them, to recognize them as they occur. Communication is critical. Communication and information are how the body is able to communicate and carry out integrated function. At the simplest level, the information is stored in the DNA, which turns into RNA and made into proteins. But we also have communication that I know you have studied a lot, signal transduction at the membrane, as well as intracellular signal receptors, such as you might have for a steroid hormone. The signal molecule that can get through the membrane then finds its receptor inside the cell. One way to think about communication is to divide it into two forms, local and long distance. Local control is short distance, and it takes place many different ways. It can go through gap junctions in the cell-cell connections. It can go, as we saw in that figure, by diffusion through the interstitial fluid. We also have contact dependent signaling, which is the way many of our immune cells work. When the immune cell meets up with the ligand and they bind together on the cell, it triggers a cascade of events. And then finally, we have the chemical signals that can go slightly longer distance, including paracrine signals, meaning they're working on neighboring cells, autocrine signals that are working on the cell that secretes the signal into the extracellular fluid, and the cytokines that are released by almost every cell in the body. Oops, sorry. The long distance signaling is primarily the responsibility of chemicals moving through the blood, and these may include both hormones that have been secreted as well as cytokines. And we have a greatly increased recognition now in this post-COVID era of the importance of cytokines in physiology. And then the nervous system uses a combination of chemical signals, the neurotransmitters and neuromodulators, as well as the electrical signals that are the signals that move down the axons, the action potential. So this is the control systems of the body. And so now we finally get to our key concept, which is homeostasis. And essentially, if an organism or a body stays in homeostasis, then everything is fine. But there can be an external change, such as a change in temperature, or an internal change, which can be both caused by a pathogen, such as the SARS-CoV-2 virus, or simply something has malfunctioned in the normal physiology. Either of these can cause a change that will create a loss of homeostasis. And at that point, the organism attempts to compensate. And if compensation succeeds, the organism stays well. And if compensation fails, this is when we have illness and disease. And this is where you, as medical students, are going to be playing a role as trying to decide what has happened to homeostasis and how can you correct it. So the definition of homeostasis is maintenance of a relatively constant internal environment. And we saw this picture earlier. The internal environment is the extracellular fluid, which then is a buffer between the cells and the external environment. The concept of homeostasis was first expressed by Claude Bernard, a French physiologist and physician, but it is that definition that we just saw is more commonly attributed to an American physiologist, Walter B. Cannon. And in one of his essays, he defined homeostasis then as the maintenance of a relatively constant internal environment. Everything about homeostasis really is aimed at keeping the brain functioning. So we have a number of variables that the body monitors. And through those monitored variables, which are also called regulated variables, then if the variable moves outside of the desired range, what you read, then there is a sensor that is activated, and that is the body's attempt to compensate. So the sensor sends a signal to an integrating or control center, and the control center then sends information to some structure or structures that we call the effectors. They have an effect on the variable, and they try to bring that variable back into range. If the variable stays within range, then there is no need for the body to activate the control center. So one of the things I would like you to do is take a second for yourself, and I hope you all have a piece of paper or a device that you can type on. And so what I would like you to do is see how many regulated or monitored variables in the ECF, in the extracellular fluid, that you can think of. So I'm going to give you a couple of seconds, maybe a minute, to write down as many regulated variables as you can. All right, let's look at my list and see how many of them you were able to think about. So blood pressure is one. That's one that we all, I think about. We worry a lot about in medicine. but related to that is blood volume. Then we have oxygen and carbon dioxide levels in the arterial blood. Glucose, highly regulated if things are going well. pH, osmolarity, potassium and calcium ions, and then temperature. So those are some of the regulated variables that we think about and that you will run into during your study of physiology. So Walter Cannon was very much ahead of his time. This is writing almost 100 years ago. We didn't know about signal molecules then. We didn't know about signal transduction and receptors and all of that. We were still working very much at the systems level. And so in his essay, he pointed out some of the variables that he felt were relatively constant. One was what he called environmental factors that affect cells. In other words, temperature, pH, and osmolarity or water balance. He also had what he called materials for cell needs. The cell needs glucose for energy, sodium, calcium, oxygen. And then what's amazing is he identified internal secretions having general and continuous effects. The endocrine system was just being explained at that point and discovered. And so the fact that he recognized that there were hormones and what we now call cytokines that were being secreted and having a constant effect is quite remarkable. So when we think about homeostasis and control, there are a couple of different patterns that you will see. One problem that many students have, including my own, is that there is a tendency to think about physiological processes as either it's on or it's off. And that's not the way things work. Many different physiological processes function under what we call tonic control, meaning that it's like a radio. If you turn on a radio, then you have a volume knob and you can turn it way down or you can turn it way up and make it loud, but the volume is always on. And that is what tonic control is. So for instance, this is a figure showing you tonic control of blood vessel diameter. So there is a neuron that is normally giving a moderate signal rate, and the blood vessel then has an intermediate or middle-sized diameter. If the signal rate decreases, then the vessel dilates. And if the signal rate increases, then the vessel constricts. And so this is a case of up or down. Do I get more signal or less signal? It's not on or off, it's up and down. Antagonistic control is also very common with signals that have opposing effects. And probably the one you're most familiar with is the frequent antagonistic control by the sympathetic and parasympathetic divisions of the nervous system. And that's what this is showing here in the cardiac system. Sympathetic stimulation to the heart increases heart rate. The antagonistic division, the parasympathetic neuron, when it signals, it slows down heart rate. So we would say that the sympathetic and parasympathetic divisions are antagonistic to each other. Here's another example where both are present all the time. Again, where we don't secrete insulin and then it shuts off, and then you secrete glucagon and it shuts off. They are both present all the time, and it depends upon which hormone has the highest concentration as to whether the metabolism is going to go into an anabolic mode, burning glucose, making glycogen and fat and protein, or under the influence of glucagon, the antagonistic hormone, we see breakdown of these molecules because it is a fasted state and the body needs the energy. Another thing that is interesting to come across is the fact that a single signal can have different effects in different tissues. And Walter Cannon recognized this. We now know that this depends on the receptor for the signal, if it's a chemical molecule, as well as the intracellular pathway of the target cell. So for instance, epinephrine, we saw that very general picture of a signal changing the diameter of blood vessels, but epinephrine, the catecholamine, can have different effects on blood vessels depending upon the receptor type. So if epinephrine binds to an alpha receptor, then we get constriction, vasoconstriction. If epinephrine binds to a beta-2 receptor, then we get vasodilation. So here is one signal molecule having different effects based on the different receptor that it binds to. So we mentioned the control systems. This is much more of an engineering viewpoint, which is there is an input signal into the control system center or integrating center, because it may have input signals coming from different places. And then their control center will give you an output signal that will then trigger some sort of response. And we have two primary control systems for the body, the nervous system and the endocrine system. And it's important to remember that not everything that happens by one of these control systems is necessarily involved in homeostasis. For a reflex to be homeostatic, it must have meet two requirements. The stimulus, the yellow oval, is a change in a monitored or regulated variable. And there needs to be a negative feedback loop that shuts off the reflex when the variable comes back to the acceptable range. So you see here a graph. This is time on the x-axis and temperature on the y-axis. And the shaded part shows you the normal range of function, the acceptable range of function. And so we see that the variable of temperature, the blue line, is able to wander around within that range without triggering a response. But if it hits the border of it, of the normal range, it will then initiate a response from the homeostatic control system. And so in this case, temperature's fallen, the control system turns on and starts to heat. And then when you reach the upper limit of the normal range, then the negative feedback, the response has reached there. So the negative feedback loop shuts off the stimulus. So you've seen this before. A regulated variable then is the variable that is monitored by a sensor. Some sensors are always active. They're always monitoring. And the carotid and aortic baroreceptors that play a role in blood pressure are a good example. They go up and down and get a response each time they're functioning. But other systems are only intermittently active. They're only active when the variable goes out of an acceptable range. And an example of this are the hypothalamic osmoreceptors that monitor the body's osmotic concentration. If the concentration falls below a certain osmolarity, nothing happens. There is no activity by the receptor or the control center. But if the osmolarity goes above a certain value, then the osmoreceptor turns on and initiates the response. So feedback, we talk about negative feedback, and it's critical for homeostasis, but not all feedback is negative. There are examples of positive feedback where you have a stimulus that starts a response and that increases the stimulus, but then the positive feedback loop causes the response to get greater and greater and greater. This is not homeostatic. This is sending the body completely out of the acceptable range. And so it requires some outside factor to shut off the positive feedback loop. One part of the positive feedback loop, then an example would be the action potential, where you have the 2-gated sodium channel. Another one that's often used is childbirth, parturition, where the pressure on the cervix then is causing a greater contraction, and it's not until the baby is ejected that the stimulus stops. So remember, positive feedback is not homeostatic. Homeostasis really, as we said earlier, is aimed at keeping the brain function. And here is our list of regulated variables. But how is a regulated variable different from a controlled variable? I think very often we use the words that we say, oh, it regulates this, or oh, it controls that. And we use them to mean something about very similar. But in homeostasis, the regulated variable is quite different than the control variable. And let's look at it at an example. So here is the baroreceptor reflex. We have showing, starting at the yellow oval, increase in blood pressure causes the baroreceptors in the carotid and aorta to fire, and sensory neurons then are leading into the medullary control center, and the body then causes a decrease in sympathetic output and an increase in parasympathetic output. There is our antagonistic control, and we will then get vasodilation and decreased force of contraction in the heart and a decrease in heart rate, all of which then combine to decrease blood pressure. And then the negative feedback loop, as soon as the blood pressure goes back into an acceptable range, then the reflex shuts off. So what is controlled here and what is regulated? And so here's another way of looking at this with the blood pressure, the sensors going to the brain, and the output to the heart and the blood vessels. So the regulated variable is the variable that is monitored by the sensor. So the blood pressure then is the regulated variable. The controlled variables are the ones that are on the output side of the reflex. They are the way the body creates a response. So the controlled variables are going to be peripheral resistance and force of contraction and heart rate, leading to a change in cardiac output. So we have no sensors for vasodilation. We do not have any kind of sensor that directly monitors heart rate. What we are monitoring instead is the combination of those, which is blood pressure. So in the baroreceptor reflex, then, the body is regulating blood pressure by controlling heart rate, force of cardiac contraction, and vasodilation. There are no sensors, as we said, for heart rate, force of contraction, or vasodilation. The other thing that is very important then for homeostasis is to maintain mass balance. And this is where we think again about the body as one compartment, with the external environment being a place where substances can enter the body or can be lost to the external environment. And so you see here this jar then is having a certain level of liquid, and for that liquid level to stay constant, the input from outside must match the output. And so many times, many of our homeostatic functions are aimed at changing either input or output. Organ system functions are quite well integrated. I think we're going to see, for instance, we have multiple systems that overlap in the control of blood pressure. So we just talked about the three, the purple, the peach color, and the blue. We talked about the cardiac output is one determinant for blood pressure. The diameter of the arterioles changes resistance of the system to flow. And then the diameter of the veins can be used if there's to constrict veins and move blood over to the arterial side to raise blood pressure. But equally important are factors that are controlled by the kidneys. So fluid intake is behavioral, but fluid loss can be regulated by the kidneys. And so if blood pressure falls too low, the kidneys will shut down in an effort to conserve volume and keep the blood pressure up. So here is the another way of looking at it. This is using our reflex pathway. So decrease in blood volume means a decrease in blood pressure. And we can then have a trigger a response that is both the cardiovascular system, as well as the kidneys, as well as behavior. So thirst then becomes a behavioral factor that can help regulate the volume and the blood pressure. And then here is the opposite, too much blood volume. So we depend heavily upon the kidneys at that point to get rid of the excess fluid. Another place where mass balance is important is the control of pH, where the input side is coming primarily from diet, but also internally from metabolism. So when we metabolize our foodstuffs through particularly glucose, remember glucose goes to carbon dioxide and water, but carbon dioxide in water then turns into carbonic acid, which then turns to bicarbonate and H+. And it is H+ that is our determinant of pH. So we have input from two sources. We have an immediate response that is a passive response, which is the buffers. This is just like your chemistry class where you did pH experiments and added buffers. But then we have two systems that coordinate on the output side. We have a rapid response from the pulmonary system. So by increasing ventilation, we can blow off CO2. And then a slower response is triggered with the kidneys. So if the lungs cannot take care of it quickly, then the kidneys come to work. So the integration of system function then becomes particularly visible when we start getting into the pathologies. And understanding the mechanisms is critical for treatment. One thing you can do is treat the symptoms, but treating the symptoms does not remove the cause. And so if you understand the mechanism behind the pathophysiology, then you have a much better way to try and reverse the pathophysiology because you're going after the cause and not merely the symptoms. And the example that is my favorite to use because it is so integrated across many body systems is diabetes mellitus, both type 1 and type 2. Type 2 shows the integration and disruption of metabolism in many systems, but type 1 for teaching is probably a little more dramatic, especially if you take someone who has gone into diabetic ketoacidosis. So this is our picture. This is, before we get to that, this is a picture showing the integrated response to dehydration. So if we have somebody that is dehydrated, we've got low blood volume, low blood pressure, and an increase in osmolarity. And what we're seeing are each of the different colored bars represents a different system that is working. So the low blood pressure is being taken care of with your cardiovascular baroreceptor reflex. The hypothalamus, we mentioned the osmoreceptors there, they're going to be triggered and they're going to secrete vasopressin. There's a passive mechanism at the kidneys, which is if blood pressure goes too low, GFR starts to fall. But then there also the very complex renin, angiotensin, aldosterone system. So the low blood pressure then is going to directly cause the granular cells to secrete renin. And now what you can see is all of these lines are crossing between the columns. They're not separate columns. So for instance, sympathetic output by the cardiovascular system works on heart and arterioles, but there's also sympathetic input to the granular cells and also to the vasculature of the kidney that will help decrease the GFR and conserve fluid to keep from losing fluid in the urine. And then one of the most fun molecules, and we've learned a lot about it in this time of COVID, is angiotensin 2, because not only does it act on the adrenal cortex, but it has many other actions that are in many ways almost more important. So angiotensin 2 acts on the cardiovascular control center. It is a direct vasoconstrictor to help raise blood pressure. It goes over to the posterior pituitary and hypothalamus to cause increased vasopressin release, and it also makes you thirsty. So you have all of these different pathways where they're triggered separately, but they're all talking to each other. And so this is a wonderful example of how the different systems work together to maintain homeostasis. In diabetic ketoacidosis, here is a type 1 diabetic. So they have, we assume they have eaten a meal, but the orange bar is showing there's no insulin release. They are insulin deficient. And now all of a sudden you start to see the same thing, which is all of the metabolic problems that arise. So you can see that's in the green here, leading to hyperglycemia. But the brain is interpreting the lack of insulin and lack of glucose coming in as starvation. So you get polyphagia, excessive eating. I think you've probably learned polyphagia, polydipsia are typical, and polyuria, the triad for type 1 diabetic ketoacidosis. So now you have hyperglycemia. The glucose that's in the blood can't get into the cells, so the osmolarity has gone up in the extracellular fluid. The high blood pressure exceeds the renal threshold for glucose, and you start to spill glucose in the urine. So you then create an osmotic diuresis because the glucose staying in the lumen keeps water with it. And now the dehydration that we just saw in the previous one is leading to thirst and vasopressin secretion, decreased blood volume, decreased blood pressure. The cardiovascular system attempts to compensate, but if it's not able to compensate and nothing is done medically, then these people go into circulatory failure, which can then lead to anaerobic metabolism and lactic acid production. And now they're between the keto acids from the metabolic derangement and the lactic acids, they're in a state of metabolic acidosis. And now you start to see the ventilation response, the Kussmaul breathing, the periodic breathing that we see as they try to blow off their acids. And then we also see acidification of the urine. So the type 2 diabetics also show many problems, but this is an acute problem with the diabetic ketoacidosis, and it shows you in one quick picture how so many things can overlap and go together. So remember that changes in one system very often are going to be integrated and have a change in another system. So remembering all of the mechanisms and the core concepts then are going to help you as you go through, and remembering that these systems don't operate in isolation. So your pharmacology becomes important because it's one of the critical interventions we have, and your pathophysiology, look for the cause. What has gone wrong with the physiology? Because that is often going to be the clue to what you need to do to get your patients well. Just remember then that physiological function is integrated, and it's up to you to remember how to do it. So thank you very much for that. And if we have time, I will stop sharing. And I'd be happy if people have questions that they want to ask.
All right. Thank you, Professor D, for such wonderful session. Now that we're opening a Q&A session. Hello? All right. Now that we're opening a Q&A session, I repeat, now that we're opening a Q&A session, everyone who has a question regarding the topic that has been just delivered before may raise their hand and directly ask your question by unmuting your microphone or simply type it to the chat box. I'm also reminding everyone to mention their names and class beforehand. Also, Professor, may I ask whether you will be answering these questions directly one by one, or would you prefer that we collect some questions first before answering all of them?
Well, I'm happy to do them as they come in. I see one already in the chat that says, I got information about a diet tip that 24 hours of starvation causes the body to go into ketogenesis. Ketones are they're a metabolic product that happens when the body starts to burn fat. And they're very strong acids. And yes, the ketogenic diet is one that has been very popular in the Western Hemisphere. We call it the Atkins diet, but these are generally high protein, high fat, very, very low carbohydrate diets, and they will also cause ketogenesis. But it also stresses the kidneys to do this. So this is not something that you would want to do without checking with your physician.
Thank you, Professor. We're still waiting for another questions. Please.
I see someone raise their hand. Nida?
Yes, Professor. Good morning, Professor D. Silverthorn. My name is Sida Sabita. My name is Sida Sabita Soliha from Gamma class. I have a question regarding the feedback mechanism. Homeostasis is the maintenance of relatively constant internal environment, as explained. One of its mechanism is the feedback mechanism. My question is that, is it possible for the feedback mechanism to not end as it's supposed to be? If yes, what is the cause and its effect? For example, in men's cycle, estrogen decrease plays a role in positive feedback for follicle-stimulating hormone and luteinizing hormone, hence causing ovulation. Is it possible for estrogen to not decrease and not causing ovulation? Thank you, Professor.
So what you have given me is an example of the menstrual cycle and the hormones and the varied feedback where this is not homeostasis. This is an example of a control system, an endocrine system, but it has variable feedback. So partway through the cycle, as I think you explained, the feedback actually changes from negative feedback to positive feedback. And so this is a programming issue. If you think like computers, control systems are programmed to respond a certain way. And I like to call them integrating centers because they are able to take in signals, conflicting signals, and make a decision on what to do because of the way they're programmed. And so what you've given me is an example where, yes, it is very possible for the feedback to fail, in which case the person may not ovulate. But that is not a homeostatic control system. That is a really nice example of a control system that is not for homeostasis.
Thank you, Professor.
Well, I have enjoyed being with you today, and we're having a big thunderstorm here. I don't know, if you could hear the thunder in the background?
No.
If no one else has a question, I may get off and go before my electricity quits.
Okay. Prof D, maybe for the last chance, you can give a final resume for your lecture today to send to our student. Resume for that. Thank you.
So, did you say you would like the like a PDF of the slides?
No. Resume like the conclusion.
Oh, a summary.
Yes,
Oh.
yeah, okay.
Okay, so the summary would be: learn to recognize the core concepts when you come across them, because if you learn them in one system, then it's easy to recognize them in the other systems, and they follow the same principles. So, for instance, if you learn the factors that regulate blood flow, then when you go to study airflow in the pulmonary system, it's very, very similar. And so the knowing the core concepts will help you take a lot of very different information and put it into separate, if you will, buckets or ideas. It allows you to group things and make them much more simple.
Yeah. Okay, I think it's a nice summary from Prof D to make you all to understand physiology. And I do believe that if you can understand physiology, can understand physiology well, so I do believe you will be a good doctor next. Prof D, isn't it like that?
Yes, that's a very good summary. Yes, your physiology is important. Medicine is physiology gone wrong.
Yeah.
So.
Okay, thank you very much, Professor D. I hope you are well and the storm is not strong enough. So I hope that it's okay for you all in Austin, Texas. Yeah.
Yes. Thank you very much. It's been lovely to see you all.
Yeah. Hope to see you directly.
Yes, soon.
Person to person.
Yeah.
Okay, thank you.
Yes.
Bye, Prof D.
Bye.
Say hello to everyone. Prof D, thank you.
Thank you, Prof D.
Bye. Makasih,
Thank you,
Bu
Professor
Dini.
Silverthorn.
Ya, bye.